Okay, go ahead.
Okay, so today I'm going to be talking about assays for small molecule binding to a quorum sensing synthase enzyme. And this is just the general scheme of what I'm going to be walking you guys through today. So let's jump right into background and significance. So antibiotic resistance is really a modern public health crisis that has been around since basically the dawn of the antibiotic era itself. Penicillin was the first antibiotic that was approved by the FDA, and it was approved in 1941. And only a year later, the first antibiotic-resistant strain of Staph aureus was discovered. And since then, we've had numerous more strains discovered, and it's become a real problem for the healthcare system. So much so that the cost of antibiotic-resistant infections and the treatment thereof costs the United States about $1.8 billion per year. And the other problem that we face is that the discovery of new antibiotics has slowed down tremendously in the past 20 years, with only about 15 new antibiotics being approved since 2000. So the quorum sensing pathway serves as a really interesting potential target for new antibiotic pharmaceuticals. So the quorum sensing pathway has a number of components, including this autoinducer synthase enzyme. This enzyme creates small molecules called autoinducers, which are represented as these little yellow circles. Those autoinducers stay outside of the cellular membrane until they reach a certain concentration. Then they re-enter the cell, bind with an autoinducer receptor, and then that receptor turns on the transcription of target genes. And we could disrupt this pathway at any point and stop the transcription of these target genes. So this is the structure of that autoinducer receptor bound to DNA and an autoinducer small molecule seen here in the active site. This is a few of the various structures of autoinducer molecules. You can see that they all share this acylated homoserine lactone backbone, but what varies is the length of the acyl tail. So there are a lot of different behaviors controlled by quorum sensing, including pathogenicity, biofilm formation, and virulence. In the case of Chromobacterium violation, they produce violation, and Vibrio fischeri is known for its bioluminescence, which can be quenched by the disruption of this quorum sensing pathway. So we are really interested in being able to control this quorum sensing pathway so that we can inhibit the transcription of the genes that cause things such as pathogenicity and virulence. So our general goal to do this is to create chemical inhibitors for the autoinducer synthase enzyme so that the bacteria cannot produce any of the autoinducer molecules and therefore cannot commence with transcription of those target genes. So this is the mechanism of the AI synthase to create these autoinducer molecules. So what we start with is this molecule over here called S-adenosylmethionine. I'll refer to it as SAM from now on. SAM reacts with acyl ACP, and we lose thio-ACP as a product. And then we have this transition state. And this transition state is really important because it can help us inform the design of our inhibitors, and it has. And then once this ring is closed and we lose this group over here called methylated thioadenosine, we have our autoinducer molecule over here. And so we have taken this transition state, and it has inspired the structure of our inhibitor. And this inhibitor is called JKR. We can see that it is structurally very similar to the transition state of the autoinducer synthase mechanism, and that's why we think that it is potent at inhibiting, I don't like those words. That's why we think that it acts as an inhibitor to these AI synthase enzymes. And it does. We have shown that 1 micromolar of JKR reduces the efficacy of CEP-I, one of these autoinducer synthase enzymes, by about 50%. And so you can see all this inhibitory effects with JKR. So we know that it inhibits it in some manner. My goal is to find methods that can confirm that JKR binds to an AI synthase enzyme, and we can determine the mechanism of inhibition of JKR. So now I'm going to walk you through my methods and some of the theories behind them and the results that I got from them. So the first method that we tried was a method called protein-observed fluorine NMR. And protein-observed fluorine NMR, or PROF NMR, relies on the idea that when we have a protein and we introduce it to a drug, the conformation of that protein is going to change, and it's going to change specifically around the active site. And if we were to introduce a tag to this active site with something that is NMR active, such as an F19, we can monitor the change in conformation of the environment of the active site by monitoring the change in chemical shift of the fluorine NMR with and without the drug. So this is an example from the literature showing a fluorinated protein. In this case, it's called bromodomain 4. So this is the fluorine peaks of the active site without a drug, and this is it with a drug. So you can see that there is a really significant change in the chemical shift, and therefore we can assume that there is a pretty significant change in the conformation of the active site. So this is a crystal structure of one of our autoinducer synthase enzymes, and you can see that we have a tryptophan right here shown in green in the binding pocket. And what we can do is we can replace these tryptophans with a molecule called 5-fluoroindole, and we can use the chemical shift of these fluoroindole molecules, or the NMR activity of these fluoroindole molecules to monitor changes. So the general workflow for these experiments is to create a media containing 5-fluoroindole, and that requires measuring out all of the amino acids, a variety of salts, vitamins, and cofactors, and instead of tryptophan, we add in 5-fluoroindole. We then express B-1, our AI synthase enzyme, in that defined media in the presence of 5-fluoroindole, and then purify and concentrate that purified protein as you would any other protein, and then hopefully observe the changes in the fluorine 19 peaks with and without JKR. So we were able to express B-1 with that defined media, including 5-fluoroindole, but we were unable to see any peaks in our NMR. And there's a few reasons that this could be. The first reason is we don't have any confirmation of or quantitative information of how much 5-fluoroindole got incorporated into our protein. So we could have just not had a ton of 5-fluoroindole in our purified protein. But it also very well could be that our NMR is not strong enough. For example, this is the NMR like where the previous image was taken. This is a 900 megahertz NMR at the University of Minnesota, and this is our 400 megahertz NMR, which I love very dearly, approximately to scale. So it's very likely that we may not have had a powerful enough magnet to see that. So we moved on to another technique called selective proteolysis. So selective proteolysis refers to the fact that when you have a protein, again shown here with this very artistic drawing, and you introduce a proteolytic enzyme which acts as a pair of molecular scissors, it will cut that enzyme into smaller pieces over time, and it occurs at a certain rate dependent on the environment. If we introduce a drug, the drug reduces the rate of that proteolysis. So it takes a longer time for this full-length protein to get cut into these shorter length proteins. So this is an example from the literature showing that when you have a free target protein exposed to a proteolytic enzyme, you have full-length protein for about 4 time units on this one. But when it's bound to the drug, you see full-length protein for a much longer time in the gel. And so we looked for an enzyme that was predicted to cut close to our binding site. And we found a commercially available proteolytic enzyme called Lyse-C. And we were able to predict using some software that Lyse-C has a cut point indicated right here at this K that is immediately adjacent to our binding tryptophan. And so if you look over here at this crystal structure again, this tryptophan is in green, that is our binding site, and this red portion located right here, that is where Lyse-C is predicted to cut. So we were able to get some pretty good results from this experiment, and it showed that without JKR, our full-length protein disappeared basically after 5 minutes at 37 degrees Fahrenheit. But in the presence of JKR, full-length protein was stuck around until about 15 minutes, maybe even 30 minutes before getting completely lysed. So that again helps us confirm that JKR is binding to our active site and it is stabilizing our protein. Another method that we used was Enzyme Kinetics. This was the most quantitative and the most laborious of my assay techniques. So I'm going to walk you through it right now. So first I'm going to talk about the different mechanisms of inhibition and how we identify them. So initially when you have an enzymatic reaction, this is what you're going to expect. You have an enzyme and a substrate. Those two come together to create an enzyme substrate complex. Some chemistry happens at the interface and you release a product. With competitive inhibition, we have an inhibitor that binds at the active site, blocking the substrate so that the substrate can no longer react to form the product. There are certain kinetic consequences to this. In this case, the Km goes up, meaning that you need to introduce more substrate to make more product than you typically would need to, but the rate of enzyme turnover remains the same. So this is the plot that we are going to be using to determine our kinetic mechanism of inhibition, and it's called a double reciprocal or Lineweaver-Burke plot. And the thing to take away here is that in competitive inhibition, the y-intercepts remain the same, but the x-intercepts differ in the presence of an inhibitor versus without an inhibitor. The other mechanism of inhibition that we are considering is non-competitive inhibition. And that is when the inhibitor binds to somewhere other than the active site, but it still prevents the substrate from effectively binding to the active site and turning over product. So here are the kinetic consequences of that. In this case, the Km remains the same, but the rate of enzyme turnover goes down. So again, in our double reciprocal plot, you'll see that in this case, the x-intercepts remain the same, but the y-intercepts differ. So the way that we went about doing these enzymatic assays was using a reagent called DCPIP. I'll remind you that in our enzymatic mechanism, we have a thiol as a product in the form of thioACP. And DCPIP is a colorimetric reagent that goes from blue to colorless in the presence of a thiol. And so we can measure the rate of enzymatic activity by measuring the decrease in the rate of absorbance. So this is the general workflow of how I set up these reactions. In each well, we have C8ACP and SAM from our enzymatic reaction, as well as JKR, our drug, DCPIP, step I, which is the auto-inducer synthase enzyme that we have chosen for this experiment, and the buffer. In this case, it was heaps. So for each concentration of JKR tested, we would have one lane of no enzyme control, and then increasing concentrations of SAM, in this case, 0.5, 1, 2, and 4 millimolar. And then it would run against one concentration of JKR, another higher concentration of JKR, and yet another higher concentration of JKR. So for those of you playing along at home, that adds up to 45 plate wells, 317 pipette tips, and all of my brain cells. But we got some really interesting data out of it. So this is some raw data from this enzymatic assay. And the way to read this is to think that the steeper the slope, the faster the enzymatic reaction is, or the more product is being pushed out. So you can see without JKR, these slopes are really steep, especially with the higher concentration of SAM, the natural substrate. But when we introduce JKR, these slopes level out, meaning that our enzyme has been inhibited and it's not putting out as much product. So after doing some mathematic manipulations, we were able to put together this Lineweaver-Burke plot of these varying concentrations of JKR. And if you look at the equations of these lines, you can see that the y-intercepts aren't all that different. They're not exactly the same, but they're not all that different. And so when we compare this to our example of what a competitive inhibition Lineweaver-Burke plot looks like, we feel fairly comfortable saying that the mechanism of inhibition for JKR is competitive. This is another assay that we were planning on using, but we didn't get the chance given the circumstances, and it's called the Fluorescence-Based Thermal Shift Assay. So what we would do is we would take a protein, in this case shown as this little circle over here, and we would mix it in solution with a dye called Cipro orange. Cipro orange becomes fluorescent when it is exposed to the hydrophobic regions of a protein, for example, when the protein is heated and denatures. So we can measure the temperature that the protein denatures at by measuring the fluorescence of the dye at certain temperatures. So the idea behind this assay is that when we increase the concentration of the drug, we will increase the temperature at which it hits its peak fluorescence, as which we can then extrapolate to mean it has denatured. So in this example that I've pulled from the literature, you can see that as they increase the concentration of drug, it denatures at a much higher temperature compared to without the drug. Again, we were planning on doing this after spring break, but unfortunately we were not able to. So that brings me into my future directions. I just wanted to put a note on this before I got into it, that I approached this exercise as the questions that I would want to ask after I finish this project. I know that there's still a lot that I need to get done for this project that I wasn't able to achieve, given the fact that I was not able to go back to campus after spring break. But these are the questions that I think are really important to think about once this project is finished. So first, I think we should expand our compound library, and I'm sure you'll hear more about this from my friend Julian, who is actively trying to expand our compound library. And it's just a necessary step to try to find a more optimized version of this drug. So I think some really interesting points where we could potentially functionalize this molecule include at the end of the acyl tail, so it can interact more with the surrounding protein, as well as this position here. I think this position would be really interesting because it is known that if you set the stereochemistry of a molecule, it has a dramatic effect on the binding affinity. And I think if we just inserted a small functional group here, maybe a methyl group, that we would see a difference in our binding affinity. The lab has already started to do some of these projects. Like I said, Julian has been talking about expanding the compound library, but another one of my lab mates, Alec, has been trying azide alkyne click chemistry to create more inhibitors. The next thing that I think we should do is assay our compound in bacterial colonies. It's very good to know that our drug inhibits isolated protein, but I would be interested to find out how that protein, or how our drug interacts within the bacterial cell. So what we would do is we would use a recombinant strain of E. coli that expresses BMI1 and then monitor the effects of JKR in vivo. And so the way that I think we could do that is by monitoring the concentration of autoinducer that is produced via GCMS. So titrate in various concentrations of JKR and then see how much that bacterial colony how much autoinducer that bacterial colony expresses with whatever concentration of JKR. The next question that I have is about the differences between AI synthase inhibitors and AI receptor inhibitors. So most of the research done in the field thus far has been on AI receptor inhibition, but I am interested to see in the effects, the different effects of AI receptor inhibitors and AI synthase inhibitors on the transcription of these target genes. So the gene that BMI1, the autoinducer synthase enzyme, expresses is confusingly named BMI1 with a lowercase. So I think it would be interesting to monitor that transcription of lowercase BMI1 via real-time PCR in the presence of inhibitor synthase inhibitors and receptor inhibitors and see what the difference is. So in summary, the quorum sensing pathway is a drug target against pathogenic bacteria without the risk of antibiotic resistance. JKR inhibits autoinducer synthase enzymes And selective proteolysis and enzyme kinetics show that JKR does bind to BMI1 and step I. And the kinetic data from these experiments seems to indicate that JKR is a competitive inhibitor. So with that, I'd like to thank the Ulrich Lab, my colleagues, Julian and Andy, who have gotten me through all of my classes, everyone in the chemistry department who has just been an awesome time to work with, all of my colleagues and my classmates who participate in my crazy whiteboard awards and stuff like that. Chun, who is just an angel and a wizard with the instruments. She's so smart, and I ask her a billion questions every day. Of course, Scott, that's all I'm going to say. And if I had to thank everyone in the chem department who helped me out along the way, I would just read the list of everyone in the department. So, oh, and the National Institutes of Health for giving us money. So here are my references. With that, I will take any questions.